Hi everyone, my name is Clarissa Wang, and today I will be presenting my project, which is in silico analysis of influenza A proteins on its druggability and genetic variability. My project was conducted under the supervision of Dr. Shapira in the Pharmacology and Toxicology Department at University of Toronto. Influenza, which is also known as the flu, is a respiratory illness caused by human influenza viruses that infect cells of respiratory tract. There are three main types of influenza virus: type A, B, and C. The influenza A viruses are found in animal and human populations, whereas the humans are considered the reservoir hosts of influenza B and C. The type A and B viruses routinely spread in people are responsible for the seasonal flu epidemics each year, while the influenza C viruses cause sporadic, mild upper respiratory infections in children. The virus uses spike protein hemagglutinin to engage sialic acid on glycoprotein of cell surfaces for entry, and infections can lead to sudden onset of chills, cough, muscle pain, fever, and even pneumonia, which can progress into occasionally fatal results, especially in teenagers and elders. While the influenza A viruses can be broken down into subtypes depending on genes that make up the surface protein, and therefore they are divided into subtypes based on two surface proteins, hemagglutinin and neuraminidase. There are 18 different hemagglutinin subtypes and 11 different neuraminidase subtypes. Compared to only a single subtype of influenza B, multiple subtypes of influenza A have been identified, like H1N1, H3N2, and H5N1. Here, we only focus on influenza A for our project, which not only because it has varied subtypes for better analysis of genetic variability, but also its high infectivity and pathogenicity that can induce the global pandemics. The type A virus is composed of a lipid envelope embedded with major surface glycoproteins, hemagglutinin, neuraminidase, and in smaller proportions, matrix 2 protein. The inner layer is matrix 1 protein together with the surface glycoproteins surrounding the polymerase protein PA, PB1, and PB2, and 8 RNA segments encapsulated as the ribonucleoprotein complex RNP. Each RNP contains a viral RNA, a polymerase, and multiple copies of the nucleoproteins. The hemagglutinin functions as a receptor binding and membrane fusion during the viral entry into target cells, and the enzymatic activity of neuraminidase is important for cleavage of linkage between the receptors and the target cells, and release of the nascent virons. The matrix 1 protein and the nucleoprotein are associated with viral RNA and are important for viral assembly as well as budding, while the polymerase proteins are required for viral genome replication. The RNPs play important roles during the virus infection by directing the viral RNA replication and transcription, intracellular transport of the viral RNA, gene reabsorbment, as well as viral genome packaging into progeny particles. For my project, I'm responsible for analyzing surface protein hemagglutinin and the matrix 2 protein of the influenza A viruses. The rationale for targeting influenza are mainly two aspects. One is the burden of influenza, and then the need for novel antivirals. Since 2010, each year seasonal flu causes approximately 140,000 to 810,000 hospitalizations and an estimated between 12,000 to 61,000 deaths annually. However, the impact of the disease was significantly greater during the three pandemics in the past century. The Spanish flu H1N1 caused about 30 to 100 million deaths worldwide, which was even more deadly than worse. Furthermore, H5N1 avian influenza virus has potential as a global threat of zoonotic infections. Vaccination is the best way to prevent infections every year, but only about 40% of Canadian adults get the flu vaccine each year, as well as the available vaccines have varied effectiveness and tend to be an issue at the early stage of flu outbreak. Other than that, the drug resistance of the approved antivirals that are currently used in Canada arise in the current circulating strains. To simultaneously develop anti-influenza therapeutics and prepare for the next pandemic threat, influenza virus drug discovery should focus on the development of pan-influenza virus agents against a broad spectrum of influenza viruses, both seasonal and novel. The goal of this work is to utilize an in silico protein modeling software to first identify the binding pockets of influenza A protein targets and analyze the structural diversity and chemical interactions between those binding pockets and their non-binding ligands. Second, to determine the druggability of the pockets if there's no inhibitor has currently been developed, and the conservation of residues lining each binding pocket across all influenza A viral strains. Third, to examine how selective mutations of pocket residues would affect the ligand binding. And lastly, to investigate the potential therapeutic pockets for broad-spectrum drugs against currently circulating drug-resistant viruses or future outbreaks of novel influenza viruses. The project was conducted using the molecular software ICM Pro developed by Mosoft. This is an in silico protein analysis software that allows the users to conduct tours from bioinformatics to computational chemistry. The first step is to confirm the reference strains for structural analysis. H1N1 and H3N2 seem to be the ones that circulate in humans, and H5N1 seems to be also high on the list of pandemic potential based on the literature. The analyzed ligand bond crystal structures are loaded into ICM Pro and derived from Protein Data Bank, or PDB, which the PDB codes are chosen in the website polymorph. The ICM Pocket Finder function was used to identify possible binding pockets for ligand binding on the protein based on a good potential map of van der Waals interaction and determine the residues contacting ligands. Note the cutoff is 2.8 angstrom for side chains lining pockets and 4.0 angstroms for side chains lining ligands. It's because the boundary of pockets is beyond the van der Waals potential of the ligand. And then, if there's no developed inhibitor, sitemap will be used to access the druggability. For the proteins that I worked, the pockets are occupied with drug-like ligands with no need to access druggability. Next, I conduct various computational assessments for genetic diversity analysis. The protein sequences of all type A influenza strains were downloaded from NCBI database, because the goal here would be prioritize strategies for pan-influenza drugs. Then, the multiple sequence alignment was performed by using the program Kalian, a tool for rapid multiple sequence alignment of thousands of protein sequences. The core sequence structures were linked to MSA and to determine the structural similarities of residues lining the pockets, and the conservation results of each pocket were obtained after running the script in ICM. This grouping of amino acids based on their R group chemistry was used for amino acid conservation analysis of pocket residues. Based on the results of the conservation table, the ICM trimutation function was conducted to calculate the change of the Gibbs free energy of the inhibitor binding associated with the genetic variations of influenza A proteins, computationally predicting the effect of these mutations on binding of known ligands. I first select hemagglutinin to review its function, 3D structures, and inhibitor binding sites. The hemagglutinin in influenza virus A is a homotrimer protein embedded in the envelope and covers the viral surface, which binds to sialic acid-containing receptor and promotes viral entry. Each of the HA subunit is initially synthesized as a single polypeptide precursor in infected cells, and cleavage of the precursor by host trypsin-like endoprotease is a prerequisite for viral infectivity. The precursor is cleaved at position 345 to 346, which makes it into two disulfide linked chains, HA1 and HA2. The HA polypeptide forms a membrane distal globular head that contains the receptor binding sites, while the H2 forms the most of the stem-like structures that anchor the globular domain to the viral membrane. The HA represents an attractive protein target for potential drugs due to its important roles in the early stage of virus infection. The initiation of the interaction of influenza virus with the human host cells starts with the trimeric HA protein binding to the terminal sialic residues on the glycoproteins of human cells. And in the active conformation of HA, the sites of receptor binding sites at the membrane distal tip of each monomer are formed by three secondary elements: 190 helix, 130, and 220 loops, with the bases made up of conserved amino acid tyrosine 95, tryptophan 153, histidine 183, and tyrosine 195. And here we analyze the genetic variability of three pockets found by ICM-Pro that are occupied with inhibitors in these proteins, and the sialic pockets, F0045 pockets, and orbital pockets. Based off the crystal structures obtained from the Protein Data Bank, or PDB, depicting ligand binding to the proteins as shown in this slide, receptor binding sites extends towards and binds to the sialic acid of sugars on host cells receptor by hydrophobic interactions and hydrogen bonds with loops and conserved residues, leading to the viral entry into human cells and starting the process of infection. The Table 1 is showing the color coding indication based on both percent identity in the conservation table and residues in the structure shown in the 3D graphic. Specifically, light yellow represents the percent identity of residues that are smaller than 70. Light and heavy orange represent the category 30 to 80% and 80 to 85% identity, respectively. The blank represents residues with percent identity that are higher than 80. A multiple sequence alignment indicates 82.5% sequence identity and 46.2% sequence conservation among the 13 residues lining the pocket of sialic acid. As an example of antigenic drift in H3N2 viruses, most mutations are found within or proximal to receptor binding site, which can result in a preference of targeting the receptor binding site by neutralizing antibodies. However, targets for broadly neutralizing antibodies is functionally conserved, like tyrosine 95, tryptophan 143, histidine 183, and leucine 194 are highly conserved residues interacting with sialic acid receptor shown in Table 2. Work from scientists in 2019 revealed an orally active small molecule, JNJ4796, which mimics the characterization and function of broadly neutralizing antibodies, like CR6261, to block the fusion of influenza virus as an effective inhibitor. F0045 is a fluorescence polarization probe that was designed to help identify small molecule inhibitor that bind to the pockets in the HA stem. A multiple sequence alignment indicates 88.9 sequence identity and 86.3% sequence conservation among the 16 residues within this pocket. As shown in Table 4 of energy calculation associated with fixed mutations found in genetic variability analysis at the binding site of F0045. If the Gibbs free energy or DDG bind values greater than 2 kcal per mole indicate mutations that significantly penalize the ligand binding, and the energy unit is kcal per mole. And here, the change of the binding energy is significant, which may play some roles in ligand binding. For example, it is quite different when binding with histidine, alanine, and serine at position 38 as the site of glycosylation. Furthermore, binding of the antiviral drug arbidol makes hydrophobic interactions with two protomer in HA trimers, resulted in a salt bridge breaking. It induces a conformational rearrangement in the binding pockets of HA, stabilizing the prefusion state of HA, which inhibits the membrane fusion in the low pH to block viral entry. A multiple sequence alignment indicates 88.04% sequence identity and 66.67% sequence conservation among the 33 residues lining the arbidol pocket. Based on the analysis of pocket residues and DDG bind values, change of the energy between alanine and leucine at position 101 is significant, which affect ligand interactions accounted for group-specific binding of arbidol. As stated in the literature, the side chains of alanine 101 in group 2 HAs are projected inwards to block arbidol pocket, and leucine 101 in group 1 HAs have longer side chains than group 2 HA. The matrix 2 protein of the influenza A virus is a homotetramer protein. It integral in the envelope at the surface of infected cell, which forms a pH-gated proton channel. Each of the M2 subunit has 97 residues, consisting the actal domain ED, the residues from 1 to 24, the transmembrane domain TM, the residues from 25 to 43, and the C-terminal domain CTD, the residues from 44 to 97. The CTD contains a cytoplasmic tail and significant amphipathic helix for protein interaction, and the hydrophobic side chain of valine 27 constricts the actal domain, and the size of the pore increases gradually until glycine 34. In a closed conformation, 4 identical M2 units, known as transmembrane helices, stabilized by 2 disulfide bonds, to form a channel in which the histidine 37 is the pH sensor, and the tryptophan gate, tryptophan 41, is at the end of C-terminal domain. Histidine 37 senses and conducts protons via protonation and deprotonation of its imidazole side chain. And tryptophan 41 acts as a gate and is locked by the intermolecular interactions with aspartic acid. In the open conformation, decrease of the pH destabilizes the transmembrane helical packing, unlock the gate, allowing the conductance of protons, while the C-terminal domain remains the interactions for the prevention of tetramer dissociation. The M2 channel is activated by low pH before HA-mediated fusion, and conducted protons acidifies interior of the viron, releasing the viral nucleoproteins from M1 layer and thereby unpacking the viral genome. Its channel activity is essential for not only the viral replication, but also the modulation of cellular homeostasis. Specifically, the M2 equilibrates the pH of trans-Golgi network and the host cell cytoplasm in order to prevent the premature conformational rearrangement of synthesized hemagglutinin when transporting to the surface. Furthermore, M2 generates curvature essential for influenza virus budding and scission. Here we analyze the genetic variability of 3 pockets with the known ligands in M2 channel by using ICM-Pro. It is also an unattractive target for proton channel inhibitors, amantadine and rimantadine, inhibiting the viral replication. Amantadine, shown in the figure, is a tricyclic amine inhibitor, occluding the pore with a single molecule, which resulted in the prevention of the viral uncoating at the early stage of viral replication. It has important interaction with valine 27, alline 30 at serine 31, and glycine 34. These key residues can be mutated in naturally occurring resistant strains. For instance, F31N mutation is predominant among the resistant strains, and V27A mutation is also popular to confer amantadine resistance to the M2 channel of influenza A virus. A multiple sequence alignment indicates 4.9% sequence identity and 87.5% sequence conservation among the 8 residues lining the amantadine pockets. Based on the calculation of the change in the Gibbs free energy for the not well conserved residues, it is noticed that there is no significant change in binding energy was observed for any of the alterations at position 27 and 31, and therefore these residues likely do not affect drug ligand protein interactions shown in Table 3. For rimantadine, molecules together stabilize this closed state of M2 through binding on lipid-facing side of channel near the gate. It interacts with tryptophan 41, aspartate 44, and arginine 45, and a hydrophobic wall of leucine 40, leucine 42, and leucine 43 to lock the tryptophan 41 gate. Drug-resistant mutations that may counter the binding effect via increasing the hydrophobicity of pore or weakening the packing of helices, thereby facilitating channel opening. Based on the soft structure, drug binding causes the closed channel harder to open, while the drug-resistant mutations destabilize the closed channel, making it easier to open. A multiple sequence alignment indicate 91.6% sequence identity and 80% sequence conservation among the 10 residues lining the pocket of rimantadine. And for the positions that are not well conserved, the negative DDG bind values indicate that the mutations will not affect binding with rimantadine shown in Table 5. The anti-influenza drugs amantadine and rimantadine are not recommended for recently circulating influenza viruses because of the widespread drug resistance. S31N is one of the most mutated residues among the drug-resistant mutants, retaining the capacity of conducting protons through channel to decrease the antiviral activity by amantadine. Because S31N is so prevalent, it is important to target for drug discovery, and M2WJ32 is discovered as a potent small molecule inhibitor that even shows greater potencies of conformational switch induced by S31N mutation than the capacity of antiviral drug amantadine against the wild-type M2 protein. It contains a charged ammonium group and binds to the backbone of V27 and sidechain of N31 align the cavity of M2 for inhibition probably through stabilizing the interactions that conduct protons to shuttling residues H31, H37. A multiple sequence alignment indicates 94.9% identity and 87.5% sequence conservation among the 8 residues lining the binding site of drug M2WJ332. And in Table 7, the energy calculation associated with drug-resistant mutations at the binding site of M2WJ322 are not significant, indicating that there is no effects for inhibitor binding, which may be explained by the tolerance to substitution in the structure-activity relationship. Consequently, Dr. Shapira provided support to think of a metric to evaluate and compare all pockets analyzed for the whole group project regarding to influenza A virus. We calculated the percent of residues that are greater than 99.9% conserved, greater than 99.9% identical, greater than 99% conserved, and greater than 99% identical for each pocket. For the final ranking, greater than 99.9% conserved may be the best metric. Furthermore, it is obvious to see in the plot that the pockets of the HA and M2 proteins are not well conserved for the development of broad-spectrum inhibitors, but M2WJ332 pockets may be good candidate for novel antivirals, shown as M2-LY0 PDB code. The hemagglutinin may be a relevant therapeutic target, and its receptor binding sites are preferred target for broadly neutralizing antibodies. And however, it is found that the side chains lining sialic acid and F045 are poorly conserved with 46.2% and 56.3% conservation across 27,876 influenza A virus sequences, respectively, indicating that this site is not favorable for the discovery of pan-influenza A virus inhibitors. While the binding sites of Arbedo are relatively more conserved with less mutations than that of the sialic acid and F045, with 66.7% conservation across the influenza A virus sequences, which is a better candidate site among these 3 pockets for the development of antiviral drug against influenza A, so that the pockets of HA ligands analyzed in this study are not ideal therapeutic targets compared with other proteins in influenza A virus. For matrix 2 protein, it is a relevant therapeutic target, and the side chains lining amantadine and rimantadine are highly conserved with 87.5% and 80% conservation across 28,484 influenza A virus sequences, respectively, indicating that these sites are relatively favorable for the development of pan-influenza A inhibitors that target the M2 channels. However, the drug-resistant mutations appears due to widely used antiviral drugs. While the M2WJ332 binding site of the M2 protein is conserved with 87.5% sequence conservation, and that specifically targets S31N mutant, suggesting this site may be a therapeutic interest for inhibition of viral infection. As such, pharmacologically targeting the conformation exchange induced by S31N mutant is a promising avenue for the discovery of a broad-spectrum anti-influenza A virus inhibitor against M2 channel to address the problem of resistance. And lastly, greater than 99.9% conservation may be the best metric to evaluate the pockets in influenza A virus. For further study, we should investigate new pockets that are not occupied by developed inhibitors yet for all proteins of influenza A, and also to keep designing novel drugs that specifically target those drug-resistant mutants. This work is supported by Dr. Shapira for always providing his help and insights, as well as the help from our primary contact on a day-to-day basis, Sitayesh, for guidance throughout my project.